Hello, welcome to the second lecture on explicit memory. I'm Matt Reza. This is the lecture on the induction and expression of LTP. We are still at the section 1 of the lectures on explicit memory, but this is a part 2. We have induction and expression of LTP in this lecture, and I'm going to introduce the concept of silent synapses as well. So let's begin, but before talking about induction and expression of LTP, I want to introduce a type of glutamate receptors called NMDA receptor. So this is NMDA receptor. NMDA stands for N-methyl D-aspartate receptor. It's a ligand-gated glutamate receptors. It means that glutamate is one of the agonists of these receptors, and I talked about one of its antagonists called APV. Okay. And this is a general structure of this receptor. These are different sites. We have phosphorylation site. We have agonist and antagonist binding sites here. We have also a magnesium block site. Okay, and the difference between they have this is the pore. Okay, this is the channel itself, and they have a magnesium ion blocking this channel. And it has a very important role in the activation of these receptors. So I'm just going to talk about that. So, that's general shape and the structure of NMDA receptors. Now let's talk about how they're activated. Actually, well, when we have glutamate, okay, released by presynaptic neurons, glutamate binds to these NMDA receptors. But, they're not activated. Calcium ions and sodium ions and potassium ions cannot go through that. Why? Because we have still that magnesium ion. Why do we still have that magnesium ion? Is because the postsynaptic cell is not depolarized. The postsynaptic cell is still at the resting membrane potential minus 65 millivolts. So, but when we have depolarization in the postsynaptic cell, for example, when we depolarize the postsynaptic cell to, for example, minus 3 millivolts, 30 millivolts, so we depolarize that. What happens is that, this magnesium blockade is relieved, okay? And now the calcium ions can get in. The sodium ions can also get in. So, these NMDA receptors act as a coincidence detector because they need a simultaneous presence of both, glutamate and depolarization. So here, for example, you can see that the inside of a cell is still relatively negative to the outside, so it's not depolarized yet. So we have glutamate here, but this magnesium ion is still blocking the NMDA receptor. But here we have depolarization, and then the magnesium ion is relieved, and the calcium ions can get in. So that was just about NMDA receptors. Very short introduction to NMDA receptors and how they're activated. Now let's talk about LTP, induction and expression of LTP. I introduced the concept of LTP that long-lasting changes in the synaptic transmission, long-term potentiation actually. But before talking about how LTP is induced and expressed at the synapse, let's talk about a normal synaptic transmission. This is a synapse between CA3 neurons, pyramidal neurons, and pyramidal neurons in CA1 area. And we know from the previous lecture that this synapse is actually is in the pathway called Schaefer collateral pathway. From pyramidal cells in CA3 to pyramidal cells in CA1 area of the hippocampus. So what we have here is that these CA3 neurons, they release excitatory neurotransmitter glutamate in this synaptic cleft. And glutamate then binds to these NMDA receptors and also another type of receptors called AMPA glutamate receptors. AMPA is alpha amino 3-hydroxy 5-methyl 4-isoxazole propionic acid. It's a one-sentence line name, and I have to thank scientists who came up with a, abbreviation for this receptor, AMPA. So we call it AMPA. And these AMPA receptors, they're also glutamate receptors, but they don't have that magnesium ion. So once the glutamate binds to them, they open, and so ions can get in. So we have here, these glutamate neurotransmitters. They bind to both. They bind to both NMDA receptors and AMPA receptors, but NMDA receptors are not letting ions to go through because the postsynaptic cell is still not depolarized. But these AMPA receptors, I told you that they don't need depolarization of the postsynaptic neuron or terminal because they don't have that magnesium ion. So what happens is that they open, and potassium ions can get out and sodium ions get in, and they depolarize the cell to some extent, and that depolarization sometimes in normal synaptic transmission is not that strong enough to relieve the magnesium blockade of these NMDA receptors and activate them. Okay, so we still don't have that depolarization because we don't have a very strong presynaptic release of these neurotransmitters. We don't induce LTP. We don't use that totanic stimulation, that high frequency stimulation of these CA3 neurons. But now let's see what happens when we induce LTP. When we apply that totanic stimulation, we stimulate the presynaptic neuron with that high frequency tetanus. What it does is that it releases more glutamate neurotransmitters. So now a large number of AMPA receptors, more AMPA receptors are activated. And then what happens is that they depolarize the cell because more AMPA receptors are activated. And so we have a larger EPSP and depolarization in presynaptic terminal. And that large depolarization or EPSP relieves the magnesium ion is great enough to relieve the magnesium ion of these AMPA receptors and activate those AMPA receptors. Once these AMPA receptors are activated, calcium ions get in. So these calcium ions have very important roles. They actually activate a calcium signaling cascade by activating calcium calmodulin, a protein, a calcium modulated protein. I talked about this structure and how it is activated and its effector proteins and how it activates CAM kinase 2, for example, in the last lecture of biosignaling in the biosignaling lecture series. But this calcium calmodulin then triggers a bunch of biochemical activities and biomolecules. For example, it activates CAM kinase 2. It activates an enzyme called nitric oxide synthase. It activates a tyrosine kinase and also protein kinase C. Let's see what they do. Okay, for example, this nitric oxide, it does a very important job in expression of LTP. What it does is that it synthesizes nitric oxide, NO. And that nitric oxide then acts as a retrograde messenger. It gets out of this postsynaptic neuron, it gets to the presynaptic terminal, okay, and tells the presynaptic terminal to release more neurotransmitters by actually activating protein kinase A and C, I think. So it signals back to the presynaptic terminal, and it tells the presynaptic terminal to enhance its transmitter release. Actually, I'm going to explain it later in the second lecture that it basically gets out of the cell not as nitric oxide, but as peroxynitrate, okay, as superoxide. But here it's at nitric oxide. Next, we have these protein kinase Cs. What they do is that they cause the insertion of more and more new AMPA receptors into the spine membrane, okay? And so we have more AMPA receptors, we have a larger current through the AMPA receptors, we have an enhanced current through the AMPA receptors. This is the induction of LTP. Well, the induction of LTP is about the triggering is about those biochemical reactions which are triggered by calcium channel modulating. Sorry, I have to pause the lecture just to apologize. I pronounce nitric oxide and peroxynitrite as nitric oxide and peroxynitrate. Nitric is a Persian pronunciation of nitric. So I'm really sorry. At the time I was recording this lecture, I was thinking about the Persian pronunciation. So the correct pronunciation is nitric oxide and peroxynitrite. Sorry for that. Let's watch the rest of the lecture. Now let's talk about what happens in the expression of LTP. Well, those biochemical reactions activated during the induction of LTP, they have two major effects on the synapse in order to express the LTP. One of them I already told you is the enhancement of transmitter release by that retrograde signaling by nitric oxide. So nitric oxide is synthesized in the postsynaptic terminal. It signals back to the presynaptic terminal, and it tells it to enhance the transmitter release by activating protein kinase A and C. And then we have another change, and that is the insertion of new AMPA receptors. What happens is that these AMPA receptors which are recycled in endosomal vesicles, and protein kinase C that I showed you in the previous slide phosphorylates the cytoplasmic tail of these AMPA receptors, and by doing that it triggers the fusion, of these endosomal synaptic vesicles into the spine membrane, and by doing that it causes the insertion of new AMPA receptors, and so we have enhanced current through these AMPA receptors, in part because of that insertion of new AMPA receptors. We have also more transmitters released in the synaptic cleft, and so this is why we have LTP. So in expression of LTP is about those long-lasting and long-term changes at the synapse. We have more presynaptic release of neurotransmitters, and we have more AMPA receptors, so new AMPA receptors. So that's all about the expression of LTP. This is a very beautiful summary. Very great summary I got from the book by David Sweatt. It tells us that both during the LTP, or when the LTP happens at a synapse, when the synapse is strengthened, both the presynaptic and postsynaptic neurons or terminals contribute to the expression and induction of LTP. So, for example, and both of them are actually altered. We have some alterations in the presynaptic terminal. For example, neurotransmitter amount in vesicles is increased. Number of vesicles released, synaptic vesicles also increases. The kinetics of release, glutamate uptake, I think it is enhanced, maybe. And the probability of vesicle fusion and the release of neurotransmitters is also enhanced or increased in the presynaptic neuron or terminal. We have also some alterations in the postsynaptic terminal. For example, the number of AMPA receptors is changed because we have the insertion of new AMPA receptors, so we have more AMPA receptors. The ion flow through AMPA channels is also enhanced, and the membrane electrical properties are also altered because we have more ionic channels, we have more AMPA receptors, and we have an enhanced current through those AMPA receptors. So, of course, the electrical properties of membrane, the postsynaptic spine membrane also change. So, yeah. So both of those presynaptic and postsynaptic terminals contribute to an induction and expression of LTP, and that's a very important point. Now I'm going to explain silent synapses. They're all about the postsynaptic contribution to the expression of LTP. So, scientists discovered about the postsynaptic contribution to LTP by a new type of synapse called silent synapse. You know, they found out that when they stimulated an action potential in one neuron, that action potential actually failed to elicit a postsynaptic response in the postsynaptic neuron. When scientists investigated this further, they realized that the postsynaptic neuron only had, or postsynaptic terminal, only had NMDA receptors and no AMPA receptors. So I told you that NMDA receptors need both depolarization and glutamate. So, but they only got glutamate and no depolarization. That's why we had no postsynaptic response or postsynaptic current in the postsynaptic terminal, in that synapse. And that's why those synapses are called silent synapses, you know, because we have no EPSP or EPSC in those synapses, at those synapses. And so let's show you a diagram of that. So this is a silent synapse I told you about. It has only NMDA receptors. So when we stimulate this presynaptic terminal, it fails to elicit an EPSP or EPSC in this postsynaptic terminal because it only has NMDA receptors and is not depolarized, okay? This postsynaptic terminal is not depolarized, so these NMDA receptors are still blocked by magnesium ions, so they cannot actually let the calcium ions or sodium ions get in. But this is a non-silent this is a non-silent synapse. It has both NMDA receptors and AMPA receptors. And what scientists did was, they next tried to unsilence a synapse, okay? And they did that by pairing a silent and a non-silent synapse together. So let's see what happens. You know, they tried to induce LTP at the non-silent synapse. That strong high-frequency tetanus, you know, released a great number of, large amount of glutamate. Glutamate is then received by glutamate receptors at both non-silent and silent receptors, because we still have a glutamate receptor NMDA at the silent synapse. And then a large number of AMPA receptors are activated at the non-silent synapse. And a large number of AMPA receptors are activated at the non-silent synapse. And so we have a large depolarization at the non-silent synapse. But you know, that large depolarization actually can propagate through the cell. So we have back propagation action potential, and that depolarization actually propagates through the cell, and it depolarizes the postsynaptic terminal at the silent synapse as well. So as a result of that depolarization, the NMDA receptors at both non-silent and silent synapses are activated. Their magnesium ions are relieved, and they're now activated. Well, at the silent synapse, once the NMDA receptors are activated, they let calcium ions get in. Calcium ions activated, as I showed you, they activate calcium calmodulin. Calcium calmodulin activate protein kinase C. Protein kinase C phosphorylates the cytoplasmic tail of those AMPA receptors, which are recycled in endosomal vesicles, and that phosphorylation of those, the cytoplasmic tail of those AMPA receptors, triggers the fusion of those endosomal vesicles with the postsynaptic terminal membrane of the postsynaptic spine. And now we have, new AMPA receptors at the silent synapse, and this silent synapse is no longer silent, because we have AMPA receptors as well as those NMDA receptors. So the next time, the action potential from these, stimulation of these presynaptic terminals, the stimulation in these presynaptic terminals is actually going to elicit a postsynaptic current or response in these postsynaptic terminals, because we have AMPA receptors here. So that was about the silent synapses and how we can unsilence the synapse. Let's review the concept of silent synapses. I'm going to show some experimental data. So we have, cell 1 making synaptic contacts with cell B. Some of the synapses that cell 1 form with cell B are silent synapses. Some of them can be unsilent synapses or non-silent synapses. So at a silent synapse, you can see that the action potential of which is stimulated in cell A fails to elicit a response, or EPSC, excitatory postsynaptic current, or any postsynaptic current in the silent synapse, at the silent synapse. This is before LTP, of course, when the membrane is still at the resting membrane potential minus 65 millivolts. Okay? And now we have, still we have it is before LTP, but we pair this synapse, this silent synapse, with another non-silent synapse, and we stimulate an action potential at that synapse, and that synapse actually depolarizes the cell. And now we have, a membrane potential of positive 30 millivolts, so it is completely depolarized. And now we have NMDA receptors, activated. They're activated, and we have EPSCs, NMDA receptor excitatory postsynaptic currents. We have a current through the NMDA receptors as a result of that activation of presynaptic neuron. Okay, and then after LTP, even at the resting membrane potential minus 60 millivolts, we can still get some EPSC, but here this time we have EPSCs. We have currents through AMPA receptors, because once these NMDA receptors are activated at the non-silent synapses, we have the insertion of new AMPA receptors, and now we have AMPA receptors at the silent synapse, so the silent synapse is no longer silent. And this is what happens. This is before LTP. This is still a silent synapse. We have only NMDA receptors. When they pair this silent synapse with another non-silent synapse, what happens is that it is unsilenced, and now we have new AMPA receptors in this spine membrane. Okay, so that was just an introduction to NMDA receptors, to very short introduction to the expression and induction of LTP, and just an introduction to the concept of silent synapses. Thank you so much for watching this video. I hope you enjoyed it. I hope you can watch other videos. Actually, the next video is the main video, the main lecture actually in the whole series, because I'm going to talk about late and early LTP, and I'm also going to talk about how LTP contributes to the formation and also consolidation of memory at the hippocampus and in other areas of the cortex. So thank you so much for watching this video, and goodbye.